This is Laura Fulford welcoming you to this Bite-Sized Bio web seminar, which today is sponsored by Leica. Leica Microsystems is a market leader in microscopy, providing wide-field, stereo and confocal solutions specifically for biological research. This year, Leica proudly celebrates 10 years of pioneering super-resolution innovation. Leica are committed to educational initiatives that provide application and technical know-how through media such as webinars and via the Leica Science Lab Knowledge Portal. Today's presentation is titled How to Pick the Right Microscopy Technique and is being presented by Dr. Alison Dunn, the Facility Manager of the Edinburgh Super Resolution Imaging Consortium at Heriot-Watt University in Edinburgh. She graduated from the University of Sussex in 2007 with a degree in molecular medicine, after which she worked at Unipath for a year in prenatal diagnostics before beginning her PhD at the University of Edinburgh. During her PhD, she used a range of microscopy techniques to study membrane dynamics, specifically exocytosis. In February 2013, she worked with her supervisor to establish the Edinburgh Super Resolution Imaging Consortium. This provides a platform for researchers to access super-resolution microscopes. She still carries out some of her own research, but primarily trains new users, works with users to develop their projects, organises events, and manages the admin and outreach. As always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Alison at the end. So now, over to you, Alison, for the presentation.
Thank you very much, Laura. Yes, so as Laura said, what I'm going to do today is talk to you about choosing the right imaging technique dependent on your scientific question. I'm the facility manager here at the Edinburgh Super Resolution Imaging Consortium, ESRIC. So this is an open access microscope facility. It's based within Heriot-Watt University and Edinburgh University. So I studied my PhD here and I looked at membrane dynamics and intercellular processes. And throughout my PhD, I used a lot of different microscopy techniques, from diffraction limited microscopy through to super resolution, and also quite a bit of single photon counting microscopy as well. So my research was predominantly on cells cultured on glass. So for this webinar today, I am going to focus really on the light microscopy techniques that are compatible with this sample prep, rather than those techniques which you would use to look at, say, tissues or whole organisms. So those are techniques like multiphoton or light sheet microscopy. So just to give a bit of an overview, we're going to look at the main questions that would be asked by a biologist. Then I'm going to work through each technique, and this is going to be dependent on the resolution that you would require. And then we're going to talk about some of the very important considerations before you even think about starting your microscope experiment. Just to let you know that the images that I'm going to show today have been acquired on our systems here at ESRIC. So you can find out about our open access facility via the website, which is at the bottom of the page. At the website, we have a tab called Your Experiment. So the flow diagram that I'm going to use today is here. It's interactive and you can work through it via the website if you'd like to. First thing that you want to establish is what is your question. Are you interested in where something is located? Or are you interested in how something is behaving? So is it moving quickly or is it moving in a random motion? Or you may want to look at interaction, so find out if two things are interacting at the intermolecular level. One tip I would give you is that I would always start with the basics. So use either a confocal or an epifluorescence microscope. And this is to get an idea of where your object is or how it's moving just on one of these systems. So if we look at these two systems in particular, if you want to have a look at, say, information in 3D, you want a nice sharp image, you want something that you don't have to post-process, then I would use a confocal laser scanning microscope. So that's just what's shown here in this movie. This is a cell in 3 dimensions. If your sample is photosensitive, I wouldn't necessarily use a confocal because it is quite slow. Instead, I would use an epifluorescence microscope as it's a lot faster, works on the millisecond time scale. Also, if you are interested in dynamics, so you have a process which is on a quite quick time scale, so less than a second, then I would also then use a microscope there. So what the movie here is showing are some vesicles which have been tracked using an epifluorescence scope, and they've been tracked using some tracking software. If you do choose to use a wide field microscope, then you can deconvolve your data. It's not a necessity, but it does help to restore your image, so it will help make it less blurry. It does this by reallocating the outer focus light, and it gives you a nicer image. It is important, if you are going to use deconvolution, that you correctly acquire your data on the microscope, and that you correctly deconvolve it, and that you're aware of any artifacts that it can produce when you do post-processing. Okay, so once you know what you're looking for, then you want to think about the scale. So how big is the object of interest? So the resolution on a fluorescence microscope is around 250 nanometers in the XY. Put very simply, what this means is that only objects that are larger than this can be resolved as a single object under normal imaging conditions. An example of structures that can be visualized as their true size under diffraction limited microscopy, so any kind of conventional scope, are large dense core vesicles, as shown in this image here. So these are found in neuroendocrine cells normally. They are between 350 and 400 nanometers. If we go down to smaller structures, so this is the actin cytoskeleton in a human pancreatic cell. So these are closer to about tens of nanometers in diameter, so they cannot be resolved under conventional scopes. And there is a lot of hidden information under the diffraction limit, but there are new techniques which can help you get closer to the true size of the structure. So that's what this image actually is. It is a super resolution image. So you get more of an idea of the network of the actin fibers. Then we go down to the level of single proteins. So they're obviously even smaller. They're nanometers in size. But again, as you can see from this image, there are techniques which allow you to localize where these single molecules are. When you choose your technique, you need to think about the resolution that you need. And the resolution you need is going to depend on your structure. And it's also going to depend on the amount of information that you require. Super resolution techniques have given amazing insight into cellular processes, but they are not always suitable for your question. Something that is really important is that you realize that you are going to learn the most by selecting the right technique and not necessarily the advanced or the most fancy one. Okay, so we're going to start with localization. So I've broken this down into 3 scales. We've got above 250, which is your diffraction limited scale, between 250 and 40, and then below 40 nanometers. And these have been divided up because they're relevant to the resolution that's offered by each technique. So I do want to note throughout this flow diagram, the microscopes that are referred to are specific to the systems we have available at ESRIC. Of course, there are other systems that can be used for the techniques that I'm going through. Diffraction limited imaging, you've got the choice between, we've got a confocal laser scanning microscope or a wide field microscope, and deconvolution being an option for either of these two. As I've already said, wide field you will get less bleaching, but with confocal then you'll get a much sharper image straight off. There is another technique available at this resolution, and that is called TIRF. So we're going to go through that now. TIRF stands for Total Internal Reflection Fluorescence Microscopy. And what happens with TIRF is it's performed on a wide field microscope, and as we can see from the diagram here, normally with a wide field the laser goes straight through the sample, it illuminates all of the sample and all of the light's collected. With TIRF, the laser light is totally internally reflected at the glass interface. So what this means is that only a very thin section, which is close to the glass, is going to be illuminated. So if your cells are really well stuck down, this means that only up to about 100 nanometers above the plasma membrane is going to be imaged. So if we look at these 2 images, we've got one wide field image and then we have a TIRF image. And the TIRF image, basically it's as if you're lying on your back looking up at the cell from below, and that's the kind of image that you get. These are some plasma membrane proteins which we can see specifically on the plasma membrane. So you would use this technique if you're interested in processes of the cell membrane within about 100 nanometers. The advantages are that you get very high contrast images, and this is because you've got a low background, because you haven't got all of the outer focus light that you would have in a wide field. Also, this technique is wide field based, so you're going to get nice fast acquisition. The main requirement with TIRF is to do with the refractive indices mismatch. To be able to create total internal reflection, you have to have a refractive indices mismatch between your lens immersion and your sample. What you really need, ideally, is an oil lens, and then you need a sample which is an aqueous solution, so in PBS or in media. Bearing this in mind, it's also important then that your cells stick well to your glass and they give a really nice footprint. The main limitation with TIRF is you don't get any Z information. So TIRF is an excellent technique which I used a lot to study membrane dynamics. It is used a lot in the field. It's compatible with many other techniques, including single molecule localization techniques, electrophysiology techniques, and photoactivation techniques, cage calcium or FRAP. Okay, so we move through the flow diagram and go down to 250 to the 40 nanometer scale. For this, you could use either a SIM or a STED, and these are both super resolution techniques. So if we start off with the SIM, SIM stands for Structured Illumination Microscopy. It's a wide field based technique, and it uses diffraction gratings to generate a higher resolution image. So what we have here are some pancreatic human cells that have been stained for actin and also for vesicles. So we can see neuropeptide, GFP, or cherry even in there. You would use this if you were looking for a moderate increase in resolution, but in all three dimensions, so in X, Y, and Z. We can get about 100 nanometers in XY and about 300 nanometers in Z. So it's not quite as much of an increase in resolution compared to other techniques, but big thing about SIM is that you get that Z increase in resolution. Also with this technique you can do multicolor. So most commercial systems can do up to 4. There are minimal requirements really for SIM. One quite important thing is that your fluorophores do need to be bright and stable. And the reason for that comes in with the limitations. So it's a very slow technique, and for each super resolution image that you acquire, you need to acquire 9 to 15 images. And between each image, a diffraction grating needs to move. So it makes it slow, it makes bleaching a bit of an issue, and so that's why you do need to have bright or stable fluorophores. SIM systems are extremely complex in their optical setup and also in their analysis, but their systems that are out there are most often turnkey systems. And so as a researcher, the images are really easy to acquire, and the reconstruction is also very easy, and that's done all through the software. So it is a very powerful technique. So secondly, in this scale, we have STED. So STED stands for Stimulated Emission Depletion Microscopy, and this uses a laser in the shape of a donut to deplete fluorescence around a central spot. The image you see here, again, we've got some actin. It works really, really well on actin and microtubules. And in one half of the image, you can see the confocal, and in the other half, we can see what happens if you have the depletion on. So we see a lot more information. You'd use STED if you wanted a resolution between about 70 and 40. 40 nanometers really is the maximum you can go with this system. The one really great advantage with STED is that you get a true image. So there's no post-processing. You increase your resolution by physically changing the size of your fluorescent spot. Also, so there's a lot of developments going on with STED. There is now 3D STED, so you can get increased resolution in Z. There are more laser lines available, which allow you to image more colors. Also, if you are used to doing deconvolution on your samples, then there is a module for STED to be able to do this. And one quite key additional advantage is that the resolution can be tailored by manipulating the power of your depletion laser. So this is something that not many other super resolution techniques can offer. So you're always going to get the best out of your STED data if your object of interest is either stripy or spotty. So microtubules or actin or something like protein clusters. Diffuse structures are not going to be resolved that well. You're not going to get a great increase in your resolution there. Another requirement is that you do need to stain with specific dyes, as these need to be able to be depleted by your laser. But there is lots of literature out there which helps you decide which dyes you can use. So the limitations with STED are usually just to do with where your depletion laser line is, because where your depletion laser line is will determine which colors you can use. So for example, if you've got a 592 depletion, so you can see that in this graph here, then really you can only use colors which are within the green or yellow spectrum. If you use gated STED, then you can't really go into the blue range, so you're limited to the green-yellow spectrum for fluorophores. So another limitation is that the STED depletion laser is very powerful. It's a very high wattage laser. It's a confocal-based technique, so live cell imaging can be quite challenging just for those reasons, but it is possible, you can do it. STED has provided great insight into protein and lipid organization at the nanoscale level, and this platform is being constantly developed to allow integration with other techniques. The system is also a turnkey system, and considering how complex the optics that are involved are, it's actually very easy to use. The ability to adapt the power of your laser and therefore your amount of resolution to your sample is unique and a very good tool. Okay, so still in localization then, but we're moving down to below 40 nanometers. And so for this, you use single molecule localization techniques. And in this diagram here, we're just going to look at the fixed techniques, as we'll go through the techniques for live samples in dynamics. So this can be broken down into either endogenous or exogenous labeling of proteins. And we're going to start with the endogenous. And for that, we would use direct stochastical optical reconstruction microscopy, which is DSTORM. DSTORM is a single molecule localization technique, and it works by turning off lots of fluorophores temporarily, therefore decreasing the density of fluorophores that are active, and it allows you to localize single molecules. So as you can see here in the movie, as molecules are turned on, they can be localized, and you can see underneath then I have a map of the localization of the molecules. So you would use this technique if you were interested in the arrangement of your endogenous proteins at the single molecule level. STORM does give amazing resolution, and it provides a map of where your molecules are. So you can get multicolor images. You can do this on a turf microscope, so you can look at proteins at the plasma membrane, or you can, there are systems now, commercial systems, which can give you single molecule resolution in the Z axis. So as this technique is antibody-based, then you do require a good antibody for your protein. You also require a specific buffer to be able to turn these molecules on and off. This buffer is specific to the certain antibody dyes that you use. But again, there is a lot of literature out there that will help you choose which one is best for you. As you can see from the movie, we've got molecules coming on and off over time. So to be able to create an image, a localization map, you need to be able to localize these molecules with a very high accuracy, as we're on the nanometer scale. So for this, you do need very good localization algorithm. Limitations with this technique. Biggest limitation with this technique is that you are using antibodies. Antibodies can create a background signal. Also, many labels will actually attach to your protein. So your signal that is created is much larger than your single protein. And when we're looking at the nanometer scale, this can make a significant difference. One of the main issues with antibodies in localizing single molecules is that they are localized more than once. So they are switching on and off reversibly in this movie that you're looking at now. Also, you're going to have more than one antibody attached to your protein of interest, and these will all be reversibly switching on and off. So just in terms of localization and quantifying that number of molecules, it's a very difficult thing to do. Okay, so if we move on to the exogenous proteins now. So photoactivation localization microscopy, or PALM, is a technique that we use to do this. It's a similar technique to STORM, but instead what happens is that you switch on a subset of molecules. They are on for a period of time, and then they are switched off permanently, so they don't come back on again. What you do then is you activate another subset of molecules, image them for a certain amount of time, and then they are switched off permanently. And you keep doing this in cycles, and that's what we're looking at in this movie here. It's like pulsing. And then what you're able to do is create a molecule map, similar to what you do with STORM. And in this image here, we can see the diffraction limited compared to our super resolution map, and see the amount of information that you gain. You'd use PALM if you were interested in where your exogenous molecules are localized and how they're arranged. Same as STORM's, really amazing resolution, about 20 nanometers. And the big advantage with this is that you only localize these molecules once. And the reason why is because you're using photoactivatable proteins. So these are fluorescent proteins which have been mutated to be selectively switched on and then switched off permanently. So your requirements are that you need to use these proteins. The best one to use is photoactivatable cherry. Same as STORM, you do also need very good localization algorithm to be able to create this kind of map from your raw data. PALM is limiting in that its single molecule dual color is challenging, as photoactivatable GFP is harder to use in combination with photoactivatable cherry. You can image in dual color quite easily if your second color is imaged under diffraction limited microscopy. So that's in the example here, where we have some large dense core vesicles in green, and then we have our single plasma membrane proteins in red. Single molecule localization methods can seem quite daunting, but there are many systems that are now available that are turnkey, and as long as you have the right sample prep and you have the right acquisition parameters, then you can achieve really great resolution. And as you can see from the images, the information that you require is so far beyond that given under any diffraction limited microscopy. Moving on to dynamics. With a resolution above 250, you've got the choice again between a wide field or a confocal. The most important factor here is the speed. If you have processes which are on the millisecond timescale, then I would go with either wide field or TIRF. If we move down to the range between 250 and 40, and then we have the SIM microscope. The SIM microscope is quite slow. So images are possible about every 600 milliseconds. So this is really closer to the second time frame. So moving down to 40 nanometers, we're looking at the dynamics of single molecules. And we're going to start here with the SPT palm. The single particle tracking palm, SPT palm, works in the same way as palm, but the difference is that you are using live cells. So you're going to be tracking your single molecules that you've activated before they're switched off. So you would use this technique if you want to track your single molecules in live cells. It is highly informative, very high resolution, and you are getting temporal information. That's the big key advantage with this technique. The requirements are similar to palm in that you need to overexpress your protein. Again, PA cherry really is the best one to use. You do need a fast camera, definitely, to be able to track your proteins, especially as they're most likely going to be moving on the millisecond time scale. Also will, again, need a good tracking and fitting algorithm. The biggest limitation with SPT palm, biggest challenge more than limitation, is the signal to noise. This is because from each molecule in your frame, you're going to be collecting a very low level of light. So your images are going to be very noisy. And what you're going to need to be able to do is denoise this image to be able to track your particles through all the noise. There are programs out there that can help you with this. In our case, we collaborate with mathematicians here at Heriot-Watt to help us process our images, and that works really well. So an additional limitation is, for now, it really is single color only. So often for live cell imaging, you'll have to overexpress your protein of interest. And this can make resolving any dynamics of these proteins very hard, as they're going to be very dense and you can't really distinguish one from another. So the ability to track single molecules is a really innovative tool. It's one that can provide great insight into cellular processes. It does require a lot of thought in the acquisition and in the analysis, but it's really worth it to get the level of information that you can get. Okay, so if we just move on to the other single molecule technique that we have, which is a single molecule detection technique called FCS, which stands for fluorescence correlation spectroscopy. In this technique, single molecules are detected as they move through a focused laser beam. So you would use this technique if you're interested in the real intricacies of how your molecules are moving, their diffusion rate or their concentration. It's a highly quantitative technique, and it's giving you this information at the level of single proteins. Requirements for it are that you need to have a low-level expression of your protein, so crippled promoters. Also, you need to calibrate your system using fluorescent dyes or proteins, and this is very important before you do any imaging. The big limitation with FCS is that there is no spatial information, but you will acquire a lot of information. It's just in the form of graphs and numbers and lots of things to analyze, but the information you get out is really good. So the last part of the flow diagram we're going to look at is interaction. This is of course only really relevant below a resolution of about 40 nanometers, as interaction happens within a distance of about 10, and I'm just going to focus on the confocal based techniques that I have here. So starting with FCCS, stands for fluorescence cross-correlation spectroscopy. So it's the same as FCS except for that you have two fluorescent proteins in your sample, and what you're doing is you're detecting changes in their diffusion rate to try and correlate their behavior. So you'd use this technique if you were interested in single molecule interaction in live cells, and this can be done in biological samples or it can also be done in vitro, same as FCS. So your advantages, your requirements and your limitations are very similar then to FCS. So you get highly quantitative information at this level of single molecules. You'd need to use crippled promoters, you need to do your calibrations, and the big limitation being there is no spatial information. So FCS and FCCS are quite complicated techniques. They do require a lot of controls and calibrations to get the right information out and be able to analyze it. But the findings from these techniques can be groundbreaking. You can perform them in vitro as well as in vivo, which is a really great advantage. And what it can do is that can provide correlative information on how single protein dynamics and interaction changes compared to in vitro to when you get it inside some cells. The last technique then is FLIM. FLIM stands for fluorescence lifetime imaging microscopy. This is a technique that you would use if you're interested in interaction and you're interested in getting spatial information from your sample. Very briefly, how it works is it detects the fluorescent lifetime of fluorophores. So what this means is if two proteins with particular characteristics are undergoing an interaction, then the fluorescence lifetime of one of these will change. And you can measure that, and by doing so you can detect whether or not an interaction is occurring. You would use this technique to look at whether or not two proteins are interacting with each other. The big advantage being that you can get spatial information. So here in this image we have an intensity-based image and we have a FLIM image. The intensity-based is quite uniform, but then when we look at the fluorescence lifetime you can actually see changes at different areas of the cell, so different interactions happening in different areas of the cell. Requirements for it are that you do require a good donor acceptor pair. So you have to use fluorophores in pairs, so either GFP and cherry together or cyan and Venus together. They're two key examples. You need to carry out a good set of controls to get your donor lifetime. The main limitation with FLIM is that it is very slow to acquire one image. For your fluorescence lifetime to be accurately calculated, you need to acquire over quite a long period, and you need to collect as many photons as possible. FLIM is a confocal-based technique, so this is all happening pixel by pixel. So again, like I said, it's a slow technique and it's not great at detecting dynamic interactions. So FLIM is a very powerful technique which has been applied in many fields of research. It can now be performed in whole animals. There's also developments underway to try and apply FLIM to camera-based techniques, which will allow for a much faster acquisition and the ability to look at dynamic interactions. We've worked through some of the possible light microscopy techniques that you can apply to your scientific question. There are, of course, many others, but way too many to fit into a 30-minute talk. So before I go through the considerations that you need to have when designing your microscopy experiments, I just want to give you a reminder of how powerful a tool light microscopy is. And when it's done right, you can get so much information. So you can look at many different cell types, so neuronal cells, neuroendocrine cells, or epithelial cells. You can look in 3 dimensions and acquire really great detail. You can look in 4 dimensions. You can track organelles over time in X, Y, and Z. And also, as I've shown you, you can also look down to the level of single proteins. And all of this has provided us with some invaluable insight into protein arrangement in many different cell types. As I said, I just want to go through the considerations when designing your experiment. You need to think about your cell type. And so does it adhere well to glass? What conditions does it need to stay healthy while you're imaging? You need to think about your antibody. So have you got a good antibody for your protein of interest or your structure? Do you know what the best concentration is to use? Have you thought about blocking? Is it going to be high background? If you're using fluorescent proteins that you're going to express inside your cells, have you thought about where it's going to be expressed? So is it going to be at the N or the C terminus? And if it is this going to affect your protein? Is it going to affect its folding or its localization? You need to think about what controls that you need, and I will always say that it is best to test out your controls first rather than leaving them till the end of the day to find out that it hasn't actually worked. You need to think about what media you're going to use. Think about if you're using media for live cells. Phenol red can cause autofluorescence, so you might want to use clear media. Think about matching your oil medium to your sample medium, or in the case of turf, not matching them. I think it's important to think about what you expect to see. So do some reading beforehand, see what's known about what you're looking for, and think logically about what you're going to expect to see. And the last thing is think about how you're going to analyze your images. So this is really important, and it prevents those times when you finish for the day and pack everything up and realize that if you'd just got one last image, then you could have found out even more. Thank you very much for listening. If you do want to get in touch at all, then you can contact me via the website, but any questions now would be great. Thank you.
Thanks, Alison, for that wonderful talk. We've got time for questions now, so if anybody has any questions, please pop them in the questions box and I'll pass those on to Alison. We've had some questions already, Alison, so I'll just start those now. So the first one is, during the talk it was mentioned several times that we need a good algorithm. Where can we find these algorithms, and how is it possible to know if the algorithm we are using is good or bad?
You can get quite a few plugins on ImageJ. There's one called Quick Palm, which works quite well. There are also some which you can get which are more based on MATLAB, and that's something if you want to get in touch with me separately, I can help you with. We did that through a collaboration, so it's not something I can directly just give out, but it is something that if you get in touch with me separately, we could do. So there are plugins out there that can do this. It is quite hard to know sometimes if it is right or not in a way. Sometimes the biggest thing is making sure that you're not picking up too much background, and I think that's just something that you can tell when you do it, really. So with the localization, was kind of the first step of the process of doing the analysis, then what I would say is make sure that, so you're collecting the actual fluorophores, and sometimes it might mean you don't collect all of them, but as long as you definitely don't collect the background. So sometimes you might have to have your threshold a bit more stringent than you might think you do, just to try and not pick up that background.
The next question is FRET at all applicable in live cell super resolution microscopy?
In terms of techniques, there is single molecule FRET, and in terms of super resolution, I think there are ifs with some single molecule techniques, yes. I'm not sure specifically in context what you mean, so maybe that's one that if you want to give a bit more information, I could answer afterwards online. Or get in touch again, like I said.
There's another one. This one might be one that you need to provide more image later, but I'll just pass it along. How to normalise the CLSM images. We've got fluorescent beads which are taken at the same conditions as the sample images, but they are clipped, which does make it impossible to subtract its values from one another.
I think, to be honest, this might be one that involves a bit more of a discussion. Yes, definitely I can understand if you, any kind of calibration where you've got clipped data is you're never going to do anything with clipped data. So, but I think I'd need a bit more information about what you're actually doing and the preparation and the experiment that you're running. So again, if you do want to get in touch, I'm happy to discuss that and help if I can.
For the person that asked that question, if you just pop a more detailed one in the questions box, we can pass that on for Alison to answer at a later date. Yep, so the next one is, why is photoactivatable GFP harder to use?
In our hands, we use photoactivatable cherry all of the time because it's fantastic. It works fantastically well. Photoactivatable GFP, we have had problems with, and I know other groups have as well, in that because what you do is you turn the fluorophores on with UV light, and then you image them with 488 nanometer light, and the idea being you image them, those activated molecules, until they are bleached all off, and then you reactivate another subset. And what happens with photoactivatable GFP is that it is actually activated slightly by 488. So you're never getting rid of all of those molecules that you've activated. So that's the real problem with it, and that causes big problems with localization, because great thing about PALM is that you localize molecules once compared to STORM, and with photoactivatable GFP you get that problem, and it means you are going to localize molecules more than once, which then isn't as good as it could be.
Okay, and how long do you need to acquire PALM data for?
It's very dependent on your sample, and it's dependent on the density of your proteins that you have inside your cells, but we often, so we look at membrane proteins, so normally it's for about 20 to 30 minutes for one cell, which might seem like quite a long time, but it's trying to make sure that you get all of the molecules, as many of the molecules as you can, to be able to form your conclusions from your data. With doing that, the biggest thing with single molecule imaging is drift. It's really, really important that you minimize drift as much as possible on your microscope, especially in situations where you do have to image for quite a long time.
What about photoconvertible proteins? What are the pros and cons of using these?
I haven't really talked about those much, and that's really just because we don't use them much here, but I have dealt with people that do, and they're very useful proteins to use. The main kind of limitation with them is that because usually they convert from green to red, and so you're using up two colours inside your spectrum, so it does mean you're more limited in dual colour. But it is very good in looking at tracking and things like that.
Most of the techniques discussed today are for adherent cells or tissues. Which techniques mentioned could be applicable for moving or flagellated cells?
I think FCS possibly, for cells that actually are moving around. I suppose if you're looking at dynamics, then I'd say the more kind of wide field based techniques. But it is difficult because if they're not adhered to the glass, then super resolution is very hard, mainly because the working distance of a lens that you use for super resolution, when you're normally using about 100 times X lens, your working distance is about 100 microns, so you're quite limited in that way. So I'm not sure how many of them would be. to be honest.
The next question is slightly cryptic, but I think they're just saying, what about FRET and FRAP? So I think they're just wanting a bit more information on those two techniques, if you can give it at all.
So FRET being the measurement of the transfer of energy between the two proteins. We generally here, we always measure that using FLIM because we find it's the best, the most quantitative. We do also have a setup here to be able to do intensity-based FRET measurements. So that again is something that you can do. The reason we do FLIM is because there's just less problems with it. Intensity-based FRET involves a lot more controls and is a lot more variant dependent on cells that day and your experiment that day and the temperature and all different things like that. And then FRAP is, I really like FRAP actually as a technique, and you can do FRAP on either the confocal and I think it was kind of started off on the confocal because you could specifically zoom in on a small region, but you can also do it on wide field or turf-based microscopes, which have it integrated, so you can still actually shoot very small regions within a cell. And the great thing then about doing it on a wide field is that you've got the speed. So FRAP is a really good technique if you want to look at dynamics and diffusion. Even just in a way, kind of, you can use it in a photoactivatable way as well. So you could use photoactivatable or convertible proteins, use a FRAP device to actually turn on those proteins. So having a FRAP device actually integrated into a system can be really great for any kind of photoactivation as well.
Can we use turf with a wide field fluorescence microscope setup, and if so, how?
So if you have a wide field microscope, you do need to have turf integrated into it. So to be able to perform turf, you need to use a combiner which allows you to hit at a certain angle, and that's something that you do within the software. Well, I know on the Olympus system we have, that's what you do. That might be a question for a physicist, possibly, who can build them. But I can try and answer that a bit more on the website afterwards. I'll ask a physicist what they say.
That's fine, but your feeling is that that's possibly
I don't
not
think so. Yeah,
not possible. That's great. Thanks again, Alison, for a very illuminating presentation and a great discussion. And thanks also to our sponsors, Leica. And finally, thanks to the audience for taking the time to attend and listen in today. If you've enjoyed this webinar and would like to view the video recording of the session, this will be available on our webinars page on bitesizebio.com. It should be available within the next 24 hours and it's free to view. There you can also see the other webinars that we have lined up in our Bite Size Bio Webinar Festival. And all it leaves me to say is, until next time, good luck in your research and goodbye from all of us at Leica and Bite Size Bio.